In the summer of 2023, a marine scientist named Andrew Sweetman sat in his office at the Scottish Association for Marine Science and looked at 10 years of data that did not make sense. Sweetman leads the seafloor ecology and biogeochemistry research group, and for a decade he had been sending instruments to the bottom of the Pacific Ocean to a region called the Clarion-Clipperton Zone, a vast abyssal plain 4,000 meters below the surface between Mexico and Hawaii, to measure how organisms on the seafloor consumed oxygen. This is standard deep-sea science. Organisms breathe, oxygen goes down, you measure the rate of consumption, you learn about the ecosystem. The instruments are sealed chambers that land on the seafloor, enclose a volume of water and sediment and the organisms living there, and then track how the oxygen concentration inside the chamber changes over time. In every deep-sea study ever conducted before Sweetman's, the answer was always the same: oxygen goes down, organisms consume it. The rate at which it goes down tells you about the metabolic activity of the seafloor community. This is so consistent, so fundamental to the understanding of deep-sea oxygen dynamics, that a result showing anything other than oxygen declining would be automatically attributed to equipment malfunction. What Sweetman's instruments had been recording instead was oxygen going up, not slightly up, not by a measurement error's worth. In some experiments, the oxygen concentration in the sealed chambers placed on the seafloor had tripled over 48 hours. Every time this happened, Sweetman and his team assumed the sensors were malfunctioning. They brought the equipment back to the surface, recalibrated everything, sent it down again. The oxygen still went up. They switched to a backup measurement method that worked on entirely different physical principles. The oxygen still went up. Over 10 years of expeditions to the Clarion-Clipperton Zone, the same anomalous result appeared again and again. Oxygen being produced in a place where, according to everything that biology and chemistry and a century of deep-sea science had established, oxygen production was impossible. There is no sunlight at 4,000 meters. There is no photosynthesis. There is nothing down there that should be making oxygen. And yet, something was. The Clarion-Clipperton Zone, where Sweetman's instruments had been recording this impossible signal, is one of the most extraordinary geological environments on the planet, and simultaneously one of the most contested. It is a region of abyssal seafloor, approximately 4,500 miles long and covering an area larger than the continental United States, and its floor is covered densely, in some areas almost completely, with polymetallic nodules. These are rounded mineral concretions, typically the size of the potato, composed of layered deposits of manganese, iron, cobalt, nickel, copper, and lithium that have been precipitating from the seawater and accumulating on the seafloor at rates of millimeters per million years. A single nodule takes approximately 10 million years to grow to the size of a potato, making them among the slowest growing natural objects on the surface of this planet, and among the most ancient in terms of the cumulative time their layers represent. They are, in the language of the mining industry that has been eyeing them for decades, a battery in a rock, containing within a single nodule the same mix of metals that goes into the batteries of electric vehicles, and existing in quantities across the Clarion-Clipperton Zone that some estimates suggest could meet global demand for EV battery metals for decades. 16 companies currently hold mining exploration licenses in the Clarion-Clipperton Zone. The fate of these nodules, whether they will be left on the seafloor or vacuumed up by industrial mining machines, was being actively debated by diplomats at the International Seabed Authority in Kingston, Jamaica, in July 2024. It was into this politically and commercially charged moment that Sweetman's discovery arrived. In July 2024, Sweetman and a 16-person international research team published their findings in the journal Nature Geoscience under the title Evidence of Dark Oxygen Production at the Abyssal Seafloor. The paper reported the oxygen anomaly, the consistent, reproducible increase in oxygen concentration in sealed chambers placed on the nodule-covered seafloor of the Clarion-Clipperton Zone, and proposed a mechanism to explain it. The proposed mechanism was seawater electrolysis. Electrolysis is the process of using electrical current to split water molecules into their constituent hydrogen and oxygen atoms. It is a well-understood industrial process. It is how hydrogen fuel is produced from water and how oxygen is generated for industrial applications. What Sweetman's team proposed was that the polymetallic nodules on the seafloor were functioning as natural batteries, geobatteries, in the term coined by co-author Franz Geiger of Northwestern University, generating sufficient electrical voltage to perform seawater electrolysis spontaneously without any external power source, in the absolute darkness of the abyssal floor. In laboratory experiments, Geiger's team measured the electrical potential on the surface of individual nodules and found voltages of up to 0.95 volts on a single nodule. Just 1.5 volts, approximately the voltage of a standard AA battery, is sufficient to split water into hydrogen and oxygen. When multiple nodules were clustered together, their combined voltage exceeded this threshold. The nodules were apparently splitting seawater and producing oxygen as a byproduct in total darkness 4,000 meters beneath the surface of the Pacific Ocean, through a geological process that no one had previously documented or even considered possible. The scientific community's response to the dark oxygen paper was immediate, divided, and in some respects more interesting than the discovery itself. The paper was published on July 22, 2024, and within days it had become one of the most widely discussed scientific findings of the year, generating global media coverage and direct policy responses at the International Seabed Authority meetings in Kingston, where the paper's findings were cited in formal proceedings by council members, who were simultaneously negotiating international regulations for deep-sea mining in the very zone where the discovery was made. Co-author Jeff Marlowe of Boston University described the implications of a confirmed abiotic oxygen source in the deep sea as potentially requiring a complete revisitation of questions about where aerobic life could have originated on Earth and about where it might exist beyond Earth. He noted specifically that oxygen provides enough energy for large organisms, animals, to survive, and that a process making oxygen without sunlight could allow for larger life forms to inhabit the deep dark waters of ocean worlds like Enceladus or Europa. Sweetman himself said the discovery demanded a revisitation of the question of where aerobic life began on this planet, given that it could no longer be assumed that oxygen production was exclusively tied to photosynthetic life in the presence of sunlight. These were large claims, and they attracted large scrutiny. A group of marine scientists and electrochemists, including Anders Tengberg of the University of Gothenburg, Angel Quister-Siskar of the University of Aberdeen, and several colleagues, published a critical analysis arguing that the original study's methodology had significant problems. Their central concern was that the measuring chambers deployed on the seafloor may not have been adequately ventilated before sealing, meaning that oxygen trapped inside the equipment during its descent through the water column could have entered the enclosed chamber and produced an apparent increase in oxygen that did not reflect actual seafloor production. Tengberg and colleagues also argued that the proposed electrolysis mechanism was, in their words, fundamentally at odds with thermodynamics, that the voltage levels measured on individual nodule surfaces were insufficient to drive seawater electrolysis under the chemical conditions of the deep ocean, UFD, and that the energy calculations required by the proposed mechanism did not balance. Critically, they noted that no other study of seafloor oxygen flux in the Clarion-Clipperton Zone or in comparable polymetallic nodule environments anywhere in the Pacific had ever observed oxygen production rather than consumption. Sweetman's team has maintained that their findings are real. They point to the backup measurement methodology, an independent sensor system operating on different physical principles from the primary optode sensors that produced consistent results with the primary data. They point to the laboratory experiments in which nodules were tested in sealed containers with a chemical poison added to kill any microbial life, and oxygen production was still observed, ruling out biological explanations. They point to 10 years of consistent anomalous readings across multiple expeditions to the same zone, producing the same result with different equipment. Sweetman has also noted that the ex situ experiments, the lab work conducted at Northwestern with nodules shipped from the seafloor, demonstrated the oxygen production effect in conditions where the equipment ventilation criticism could not apply, since the nodules were in sealed containers in a controlled laboratory environment. The critics' response to this has been to question whether the laboratory conditions accurately replicated the chemical environment of the deep sea floor, and whether the poison used to kill microbial life was itself influencing the electrochemical measurements. The debate between these two positions is not, as of early 2025, resolved. The original paper has not been retracted. No independent research team has yet published a replication study confirming the dark oxygen production under controlled conditions. The scientific question of whether polymetallic nodules on the deep seafloor are genuinely producing oxygen through seawater electrolysis remains one of the most actively discussed open questions in marine science, and the stakes of its resolution extend far beyond the academic community, into the boardrooms of mining companies and the chambers of international regulatory bodies. The specific mechanism proposed by Sweetman and Geiger, that polymetallic nodules function as natural geobatteries, generating electrical voltage sufficient to split seawater, is extraordinary if confirmed, and its extraordinariness extends in both directions from the immediate scientific question. Looking backward through Earth's history, the existence of a geological mechanism for abiotic oxygen production raises a question that Sweetman himself articulated. Where did aerobic life begin? The conventional understanding of the origin of aerobic life on Earth is built on the Great Oxidation Event, the dramatic rise in atmospheric oxygen approximately 2.4 billion years ago. That is attributed to the evolution of cyanobacteria and the explosion of oxygenic photosynthesis. This timeline has been understood as the boundary before which aerobic life could not have existed, because oxygen was not available before photosynthetic organisms produced it. If abiotic oxygen production through mineral electrolysis has been occurring on the deep sea floor throughout geological history, and there is no reason to think the Clarion-Clipperton Zone's nodules are geologically unique, then oxygen may have been available in the deep ocean long before the Great Oxidation Event, in quantities and locations that could have supported early aerobic metabolisms. This would not overturn the conventional narrative of photosynthesis driving the Great Oxidation Event at the global scale, but it would open a possibility that certain lineages of life may have begun using oxygen far earlier than the atmosphere's transformation suggests, in isolated deep-sea environments where mineral-derived abiotic oxygen was locally available. This does not overturn the conventional narrative of the Great Oxidation Event, but it opens a question at its margin that was not previously considered open. And looking forward into the search for life beyond Earth, if oxygen can be produced abiotically through the interaction of water and mineral surfaces without any light, then the presence of oxygen in the ocean or atmosphere of another world is no longer an unambiguous biosignature. It might be a geological signature instead, produced by nodule-equivalent mineral formations on the floor of Europa's ocean or Enceladus's subsurface sea. The conflict of interest at the center of the dark oxygen story adds a dimension of institutional complexity that is unusual even for a controversial scientific discovery. The research team that published the dark oxygen paper received funding from the Metals Company, a Canadian deep-sea mining firm, and from UK Seabed Resources, a subsidiary of the British arm of Lockheed Martin. Two of the entities with direct commercial interests in the polymetallic nodules that are the subject of the discovery. The fact that research funded by an organization that wants to mine the nodules produced findings suggesting that the nodules perform an essential ecological function whose removal could damage the deep-sea ecosystem is, from one perspective, a validation of the integrity of the researchers involved. From another perspective, critics of the paper have noted that the funding relationship creates an inherent complexity in assessing the study's claims. and that the paper's rapid and high-profile publication, combined with its immediate policy impact at the International Seabed Authority, raises questions about the appropriate relationship between preliminary scientific findings and international regulatory decisions. The scientific debate about dark oxygen is therefore simultaneously a debate about the methodology of deep-sea measurements, the thermodynamics of mineral electrochemistry, the ecology of the abyssal seafloor, the history of oxygen on Earth, the search for life beyond Earth, and the governance of the deep ocean's mineral resources, a convergence of scientific, political, and commercial questions in a single set of data points recorded in sealed chambers on the floor of the Pacific Ocean, 4,000 meters below the ships of the mining companies that funded the work. What the dark oxygen story ultimately represents, regardless of how the scientific debate resolves, is a demonstration of the deep ocean's continued capacity to present the scientific community with phenomena that its existing frameworks did not predict and cannot easily accommodate. A decade of anomalous readings dismissed as sensor error, a proposed mechanism that challenges established electrochemistry, a finding that arrived simultaneously as a potential rewriting of the history of oxygen on Earth, a potential expansion of the concept of habitability beyond the solar system, and a regulatory challenge to the most commercially significant deep-sea mining frontier in history. The science of the dark oxygen claim will be resolved in the way that science resolves contested claims, through replication, through independent measurement, through the accumulation of evidence that either confirms or refutes the original findings in conditions rigorous enough to satisfy skeptics on both sides. That process is not completed. It may take years. The deep ocean is not a convenient laboratory, sending instruments to 4,000 meters, retrieving them, analyzing their data, repeating the experiment with improved methodology. Each cycle of this process takes time and resources and ship time that is expensive and scarce. The question of dark oxygen will be answered, but it will be answered on the ocean's schedule, not on the schedule of the mining companies waiting to extract the nodules or the environmental organizations waiting to protect them. What is already clear, regardless of the eventual scientific verdict, is that the nodules of the Clarion-Clipperton Zone are not simply rocks. They are not simply battery metal deposits waiting to be collected. They are complex electrochemical systems that have been forming and interacting with the deep ocean for millions of years, hosting biological communities on their surfaces, influencing the chemistry of the water around them, and, if Sweetman's findings hold, producing a gas that the scientific consensus had believed required light to make. The scientific consensus on dark oxygen has not formed yet. The replication studies that would confirm or refute the original findings have not yet been published. The question of whether the nodules of the Clarion-Clipperton Zone are genuinely splitting seawater in the darkness 4,000 meters beneath the Pacific is a question that, as of now, the ocean has not finished answering. But the fact that the question is being asked at all, that 10 years of anomalous oxygen readings from sealed chambers on the abyssal floor have produced a genuine scientific debate about the origin of aerobic life, the possibility of extraterrestrial oxygen, and the environmental consequences of removing potato-sized rocks from the floor of the Pacific, is itself a measure of how much the deep ocean still contains that the surface world does not yet understand.